All right, so in the last section, we kind of talked about membrane transport and the role that proteins play with that. So now just kind of talking a little bit about the transport. Diffusion, of course, is a passive form of transport. In other words, there's no outside or external energy in the form of, say, ATP that's required. So this is where you go from an area of high concentration on one side of the membrane, and then you'll diffuse down to an area of low concentration until you reach equilibrium. And this is basically what we refer to as passive transport. And if you think about it, if we have a rag soaked in something that stinks really bad, like gasoline or something like that, and we put it in the corner of the classroom or in a corner of your room somewhere in the house or the garage or something like that, eventually that smell is going to permeate the entire area because it's moving from an area of high concentration to an area of low concentration. We don't really have to add any energy to it. It's just kind of a natural phenomenon that's going to occur as a result of the concentration differences between the rag that has a high concentration and the surrounding air that has a low concentration. And if you look here at this particular type of a drawing or whatever, if we have one solute here, you can see that on this side there's a high concentration, and of course on the right side here it's low. And then through time, if this is a semi-permeable membrane of some sort right here, you can see that these things, just due to the natural energy that they have, begin kind of moving back and forth across the membrane. And then eventually, once you reach equilibrium, there's still a movement, but there's no net movement. So in other words, as many particles that move to one side of the membrane, okay, will be moving to this opposite side of the membrane as a result of just these natural molecular interactions, okay? And if we have two solutes or more dissolved in a solution, they're going to equal out. In other words, if you look here, because the concentration on this side of the purple is high, and on this side on the left here, the orange is high, after time, after we go through, some of the purple ones are going to be moving across the membrane, some of the orange ones are going to be moving across the membrane, and then eventually you'll get to a point, of course, like above, with equilibrium, and you'll have as many orange moving one way across the membrane, and then of course moving the other way, as you do purple ones moving one way as you do moving the other way. So in other words, they're just going to be mixing back and forth in equal amounts, and you've got no net movement. Net meaning you're going to have a net positive gain or a net negative loss on one of the two sides of the membrane. It's just going to be equal. Purple will move right and left across the membrane. Orange will move left and right across the membrane, but all the while on both sides of the membrane, the particle numbers are going to be equal. That's the equilibrium that we were talking about. Okay? So osmosis now, this is just simply the diffusion of water. It's the movement of water across the semi-permeable membrane. We did, of course, a couple of labs that kind of talked about this with the corn syrup and the dialysis tubing. We did it with the cells and the sugar water, with the potatoes, with the seaweed, that sort of a thing. That was kind of looking at osmosis. And when we were talking about this, we said one of the things that kind of governs how the water is going to move is this notion of tonicity. And this is the ability of a solution to gain or lose water. So if we're looking at something here, if you take and you put a cell into a hypotonic solution, okay, what's going to happen is water is going to rush into the cell, and the cell's going to swell up. That part you probably remember. And if you put it into a hypertonic solution, okay, the opposite's going to occur. There's going to be more water inside the cell relative to the hypertonic surroundings, so the cell is going to lose water. And then, of course, if we're in an isotonic environment, you have no net movement of water. You've got as much moving into the cell as you do moving out of the cell, and that's what we call equilibrium. And a couple of the things that we talked about when we were discussing this in class is just like that isotonic bag of saline that you get if you're in the hospital and you're dehydrated, or you can't have fluids because you're going to have surgery or something like that. They're just trying to match the tonicity of your cells with that of the fluids that they're giving you to kind of maintain your hydration levels. Okay, so again, isotonic, you've got no net movement of water. Hypotonic, you got more water moving into the cell than you do moving out. And with a hypertonic solution, you've got more water moving out of the cell than moving in. And you can see here with these this diagram here, you've got some animal cells that lack the cell wall, so you can see dropping the cell into a hypotonic solution. The cell will take on water and burst. Drop it into an isotonic solution, you're going to have no net movement of water. It's going to maintain its hydration level. And if you put it in a hypertonic solution, the cell's going to shrivel up. And the same thing will happen with the plant cell, but the plant cell is kind of bound by a cell wall. And so that's going to kind of protect the plant and the cell to ensure that really no damage occurs within reason. If you get too hypertonic, if the solution, you should say, that you're watering the plant with or something like that, or if the soil gets too dry, or if it's the solution is too hypertonic, then the cells will continue to lose tons and tons of water, and they'll become plasmalized and could potentially die. Okay, so it's important to know that the animal cells, of course, lack the cell walls, and then you can get lysis, cell, the bursting of a cell, or you can get the cell shriveling up because of the lack of the cell wall. But the plant cells have these cell walls, and these things are important in helping the plant kind of maintain its kind of shape. And when plants are fully hydrated, they've got a lot of water, we say that they're turgid. Okay, and you can see this word in orange here. The turgidity is kind of a healthy state of the plant. The plant has a lot of water. So when you look here, and this is kind of a picture that we took of one of the cell preps that we made, and you can see here that really all of the things around the edge, these are the chloroplasts, kind of smashed right up to the cell wall, because the interior here has a lot of water. So the plant would have a very healthy looking appearance in this case. And then if the plant cells are really dry, and in this case, or soaked in a very hypertonic solution like we did with the sugar water, the cells start to undergo plasmolysis, where they're shriveling up, and you can see everything is peeling away. So in other words, this is the cell wall kind of around this particular plant, and you can see that the cell is peeling away there because the water is leaving the cell, so the cell's actually shrinking. And when the plant gets into this state, it takes on a drooped or wilted kind of appearance, and we say that it's flaccid, which is one of the words in orange there. And so, that's when the plant, we know it needs water. So if you look here, the plant on the top, this particular one right here, this, we had gone away for a while, hadn't watered the plant, so the plant starts to wilt because it's kind of taking on this appearance here. The plant cells have lost a lot of water. And then when you pour a glass of water on it like we did, and then come back in about an hour, you can see that plant starts to take on excess water, and then it gets into this healthy state like you have here, where it's turgid, and it starts to stand up and take on its kind of healthy appearance, so to speak. Okay, so now facilitated diffusion, the important thing to remember here is the word diffusion. Okay, diffusion means passive transport, no ATP energy. So facilitated means to help out. So again, it's facilitated diffusion. There's no energy, excess or external form of energy in the ATP that's required to move things in and out of the cell. It's just going to kind of move a substance from one side of the membrane to the other down a concentration gradient with the help, say, of a protein or something like that. Okay, so it's just facilitated diffusion. It's still a passive form of transport. The two types of facilitated diffusion, you can see here in this particular case, these ions are moving, or the solute is moving freely across. It's just a channel protein that's always open and makes sure that the amount of substance on both sides of the membrane is always equal. And then the other one is where the protein is going to kind of change shape as a result of, say, interacting with these solute molecules. But still, it's a passive kind of form of transport, in that this substance here causes this protein, this purple thing, to kind of change shape. And when it does so, it kind of shuttles it across the membrane. You can see that it gets it to the inside. So you've got a channel protein or a carrier protein, but they're both not requiring any ATP at all, and that's why they're referred to as facilitated type of diffusion. Okay, so let's take a look at this video here.
In order for the cell to survive, many different materials must be able to cross the plasma membrane. When water passes through a cell's membrane, the type of diffusion is called osmosis. When solute particles pass through a cell's membrane, the type of diffusion is called dialysis. Before looking at this movement through the membrane, it is important to understand the movement of molecules in general. Atoms and molecules are in constant motion. This random motion will disrupt the cluster rather than maintain it. This motion is referred to as Brownian movement. This motion is always present in some degree, in gases, in liquids, and even in solids. For instance, if a bottle of perfume is left open in a room, eventually, no matter where you are in that room, you are able to smell the perfume. the molecules of air hit the liquid, causing the liquid molecules to move into the air. At the time the bottle is first opened, there is a high concentration of perfume molecules in the bottle, a very low concentration of perfume in the air. As the perfume molecules are bombarded by the air, the concentration of perfume is dispersed into the air. In other words, the perfume is moving down a concentration gradient, high to low. This movement is called diffusion. It uses Brownian movement. Now let's take a look at another example of diffusion. In this aquarium of water, we drop some blue dye. As the water molecules move, they bump into the dye molecules. The dye molecules, at first in a high concentration, start moving and bumping into each other and the water molecules, moving into the area of lower concentration. As this movement continues, the dye spreads out and equilibrium is attained. Equilibrium is equal concentrations of the dye and water in every area of the aquarium. Remember, Brownian movement continues even after equilibrium is reached. Although extremely thin, the plasma membrane separates the contents of the cell from the environment. The membrane has several functions. This video concentrated on the movement of materials through the plasma membrane. Remember that this membrane is selectively permeable and can prevent materials from passing through or keeping materials in, and it will permit, even facilitate, the entrance and exit of materials. Materials may move through membranes passively or actively. In this video segment, passive transport, which uses Brownian movement, was introduced. Passive transport includes diffusion, defined as the movement of materials from a region of high concentration to low concentration. Two specific forms of diffusion are osmosis and dialysis. These processes are essential to the survival of the cell. Nutrients must enter, wastes must exit. The structure of the membrane allows for the processes to take place, which enables the cell to live.
All right, so that did a pretty good job, I think, of explaining kind of some of the stuff that we went over and just kind of looking at it in a video form, reviewing, so to speak. And if you've got any questions on what they said, just ask and we can watch the video again in class if we need to. Okay, so now active transport. And active, of course, means that it's requiring the ATP molecule in order for this to happen. And the kind of classic example that's used is the sodium-potassium ATPase pump. In this pump is basically operating to keep the sodium concentration on the outside of the cell membrane high and the potassium levels low, and then on the inside keeping the sodium levels low and the potassium levels high. And you see this occurring in, for instance, like nerve cells that help transmit the electrical energy from the surroundings, the sensory input kind of through the central nervous system for processing and then coordinating a response. And in order to do that, you have to have action potentials that occur, and the action potential and the ability to generate an action potential is set up by the sodium potassium ATPase pump. And that's something we'll learn about when we learn about the human body. But the key here with active transport is that you're going to hydrolyze ATP in order to achieve the goal. So you're going to pump things against their concentration gradient. In other words, from low to high. And you need the ATP energy in order to do that. Okay, so with this particular movie right here, okay, we'll take a look at this. And this kind of explains a little bit about how this pump works.
The sodium-potassium pump is an active transport mechanism that is driven by the breakdown of ATP and works through a series of conformational changes in a transmembrane protein. 3 sodium ions bind to the cytoplasmic side of the protein, causing the protein to change its conformation. In its new conformation, the molecule becomes phosphorylated at the expense of a molecule of ATP. The phosphorylation induces a second conformational change that translocates the 3 sodium ions across the membrane. In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid. The new conformation has a high affinity for potassium ions, 2 of which bind to the extracellular side of the protein. The bound phosphate now dissociates, and the protein reverts to its original conformation, exposing the 2 potassium ions to the cytoplasm on the inside of the cell. This conformation has a low affinity for potassium ions, so the two bound potassium ions dissociate from the protein and diffuse into the interior of the cell.
Okay, so that just kind of explains how the sodium-potassium ATPase pump works to get these sodiums out and the potassiums in against their concentration gradient. Okay, so now here with this last slide, what I want to do is kind of look at another video that kind of explains some of the stuff that we've been talking about. Okay, so let's have a look.
Part of metabolic activity is the ability to move materials into and out of cells. This is important in cell communication and normal cell function. For example, in order for the cells of your nervous system to function properly, ions, water, proteins, and other molecules need to be able to pass into and out of cells. The cells of the nervous system form networks. They, like all cells, are able to function because they can control what substances are inside the cell and what substances stay outside the cell. These materials move in and out of cells by passing through the plasma membrane of the cell. The plasma membrane surrounds the cell and separates the interior of the cell from its environment. This semi-permeable membrane is composed of a lipid bilayer. Phospholipids make up a large part of the membrane and form a bilayer. The structure of the bilayer is due to the tail-to-tail packing of the nonpolar hydrophobic tails, composed of two fatty acid chains, and the polar hydrophilic heads, composed of glycerol and phosphorylated alcohol. The lipid bilayer is 5 to 10 nanometers thick and is embedded with proteins. Some cell membranes also contain cholesterol. A plasma membrane contains different types of proteins which are specific to the particular function of the cell. These proteins also enable the cell to interact with its environment. The entire structure of the plasma membrane can be described as a fluid mosaic model. The phospholipid bilayer has properties resembling fluids, and the differing proteins and their attachments on either side of the membrane resemble a mosaic. The plasma membrane is the gatekeeper of the cell, allowing certain substances in and out of the cell at certain times in certain amounts. Diffusion is a process in which substances move across a membrane from an area of high concentration to an area of lower concentration, or between areas of opposite electrical charges. This is called the electrochemical gradient. Small, non-charged particles, primarily gases such as oxygen and carbon dioxide, can diffuse through the plasma membrane by moving in between the phospholipids in the bilayer. However, the cell needs to control what enters and leaves, and so transport proteins aid in the selective movement of other molecules across the membrane without the input of energy. Through facilitated diffusion, larger molecules, polar molecules, and charged ions use channel proteins embedded in the bilayer. The transport of other substances requires a special carrier that will bind the substance on one side of the membrane, which triggers a conformation change in the protein carrier, causing it to release the substance on the other side. When the substance being moved across the membrane is water, the process is called osmosis. The cytoplasm of the cell, as well as the interstitial fluid, is composed of solutions. The solvent, usually water, moves across a semi-permeable membrane toward a higher solute concentration, consisting of various molecules or ions, until equilibrium of the solutions is reached. The plasma membrane contains proteins called aquaporins, which are specialized channels for the movement of water during osmosis. A cell in a hypertonic environment will have water move from the inside of the cell toward the higher concentration of solutes in the solution outside the cell. In a hypotonic solution, the concentration of solutes is higher inside the cell than the outside environment, so water will diffuse into the cell. When the solutions on either side of the membrane reach equilibrium, they are referred to as isotonic. When substances need to be driven against their concentration gradient from low to high concentrations, active transport is required. This process uses energy to move a substance across the membrane using selective protein carriers. The sodium-potassium pump is a specialized membrane protein carrier, common in most animal cells, that moves sodium and potassium ions across the plasma membrane. ATP fuels the pump in the movement of these ions from low to high concentrations, moving sodium ions outside of the cell where they become concentrated and bringing potassium ions into the cell where their concentration is higher relative to the outside. In other instances of active transport, the concentration gradient created from the action of one pump can be used as the input of energy to drive a different pump, a process known as coupled transport. The large concentration gradient of sodium ions is used to bring glucose into the cell against its concentration gradient. The plasma membrane, with its lipid bilayer and various embedded proteins, provides a barrier between the internal cellular environment and the environment surrounding the cell. The structure and function of the plasma membrane, coupled with the processes of moving materials into and out of cells, allow for the metabolic action cells need to function, as well as for the continuation of life.
Okay, so that served as a pretty good review of parts 2 and 3 of Chapter 7 here. So if you have any questions, make sure you write them down, and we'll talk to you soon.